Thanks to all the organizers for this great invitation. It's really exciting to be here in this great place. I'm going to talk. This will be a talk that will be. I'm going to try to hit as broad a group as possible. So there'll be a certain amount of more tutorial. I apologize for anything that people may know already in the mathematics of it, but hopefully there'll be something that everyone can kind of take away from it. Ultimately, we will get to the title of this talk, but we're going to do some introduction. And I have just about the right amount of slides for the amount of time allowed, which is very unusual for speakers. So let's see here. Okay. Anyway, so what we're most interested in to talk about here is the classification and understanding of the cell types in the mammalian brain. This is a problem which has occupied neuroscientists for quite a long time, since the realization that there were cell types in the brain and the work of the famous neuroscientist Ramon y Cajal in the early 1900s. But today, we try to profile these cells in using single cell technologies, things that were not really available before 10 years ago, more or less, 10, 15 years. And we can bring multiple modalities to this. And cell type classification is like anything, if you say, well, how many types of cars are there, right? It's kind of an ill-posed question, perhaps, because it depends on what you want to use the cars for, right? And there might be different numbers depending on those functions. And it's similar for cell types. But we do characterize, ultimately we'd like to know, I think we'd probably agree that a cell type you really couldn't become promoted to be a real genuine type until we really knew that you did something, right? There should be some functional significance to the existence of a type as opposed to just a mathematical classification. But a lot of what we know about cell types today is really a more mathematical classification, if you will. It's a characterization, and we don't understand, except for a few in some sense in the brain, what it is that they really do. And I'll talk quite a bit more about this. But in essence, so the tools that we have available to us are really kind of a set of a few different modalities. One is transcriptomics, and now we can do single cell gene expression studies in the brain. We can also do regulatory studies, right? We can understand kind of epigenetic phenomena. And we can understand physiological data, such as Professor Crook here spent a great deal of time studying the kind of electrophysiological properties and modeling them. And that's another big thing. And that's probably one of the closest things we have to understanding functional significance. And then there's also their shape. And like trees, although every tree is different, the morphological shape of neurons are very, in some sense, kind of characteristic, but there's an immense amount of variation, and the understanding that will be interesting. There's also kind of, very importantly, the connectivity. To understand cell types, you really have to understand which types are connected to which, so that one can build appropriate circuits to understand function in the brain. And we can do this through genetic tools, right? There's ways of manipulating these types to understand their function and get at this information. So, the earliest classification of this done, like as I mentioned, by the neuroscientist Ramon y Cajal. Ramon y Cajal, I can't make this joke in too many venues, so I'm going to make it here. But Ramon y Cajal is a little bit like Gauss in neuroscience. If you give a math talk, you're almost obliged to mention Gauss somewhere in the talk, right? Well, in neuroscience, you will almost never see a neuroscience talk, even today, without the name Ramon y Cajal appearing somewhere in the slide. He was this Spanish Catalonian, born in this little kind of place right here called Patilla de Aragon. And he basically kind of grew to prominence through an incredible dedication and ability to kind of understand, and he had a great artistic flair because he was able to draw using a microscope and kind of camera lucida these patterns of immense resolution, really, if you think about it, for the day. there were not high quality microscopes in those days. And this was continued into even more recent times through the Hungarian neuroanatomist János Shengatulai. And so this has been done in many different kind of ways. Ramón y Cajal was like, he believed that in fact that neurons and cells in general were discrete entities and communicated through essentially either chemical or electrical kind of contact. But he didn't really understand that at that time. And he based these drawings on a certain stain called the silver stain, derived by a colleague of his named Camillo Golgi. And Golgi was Italian, and they didn't get along at all. They even fought they received the Nobel Prize together, I think in like 1906 or something like that, and they fought even at the Nobel ceremony because Golgi felt that there was this giant reticular network in which everything was connected like a big neural net. And Ramón y Cajal insisted that there were gaps and that there was some kind of communication across these gaps. Actually, around maybe early 2000s, a group with the colleagues of ours, Rafa Yusta and Giorgio Ascoli, they held a meeting in this Patilla de Aragon in which they called it the Patilla Convention, and they sought to kind of characterize and classify a certain kind of neurons called interneurons, or these are neurons that sort of express basically this GABA, and they're involved in inhibition, right? Stopping firing patterns in the brain. And so what they did, they tried to devise criteria for classifying these, like there are different physiological things, whether the cell was bursting, whether it was essentially rapid firing, et cetera. And they used a lot of kind of proposed very mathematical type of things for the classification of their morphology, such as certain angles and tortuosity, something maybe people have heard of before called shoal analysis, where essentially you grow different spheres of different things and you ask how much is touched by the branching as I move into the radial kind of thing like that. Today, we have the techniques involved today are much more kind of high-powered in terms of understanding cell type diversity, and these include single cell type transcriptomics. We can actually do a kind of something called a tax-seq to explore regions of open chromatin. We can measure methylation, which occurs prior to a gene being expressed, and we can do this actually sometimes in the same cell simultaneously, a technique called multiome analysis. And there's ways of understanding connectivity. There's ways of looking at, here you can see, so what was called spatially resolved transcriptomics, or ways of actually doing kind of in situ gene expression, where you design a certain panel of genes and you can actually get the expression of that set specifically in a cell. When we first did the Allen Brain Atlas in 2003 to 2006, this was using in some sense, the grandmother of this technique. It was basically in situ hybridization, where you design one probe for each gene, and then you march through the whole genome, building all these kind of sections. That technique was invented around 1980, and it wasn't industrialized until more or less the year 2000. So there's lots of techniques, and some we won't have time to go into all of them, but I will mention certain other ones as we go along. This is kind of an important one here because it serves in a way of discovering kind of ground truth in our data. And it's something invented by a really strong neuroscientist who's also very good at kind of computation, and from the Baylor University, Andreas Tolias. And he and his students invented this technique called patch-seq. And patch-seq enables you to kind of do the triple kind of crown of electrophysiology, transcriptomic profile, and morphology in the same cell at the same time. And it sounds too good to be true, and it kind of is too good to be true because it actually has a high failure rate and it's very kind of slow in its output. But it is often used to kind of understand how things really link without having to do association studies, kind of computationally between them. For example, the way it works is basically you first record from the cell, you patch it with a small kind of micro pipette, and you can actually, this locks onto the cell. I know Sharon understands this very, very well, and you basically can record its physiological firing patterns from that. And then through this pipette is curious because it can take out the RNA either from the cell or the nucleus of the cell and ship that off to be kind of sequenced. And then when you're done with that, you sort of fill it with something called biocytin, and then this kind of stabilizes the thing, and it can be sliced, and then you can actually image it to get the morphology. So now you have a way of, you have a Rosetta Stone in some sense linking these three. And whereas you can do hundreds and thousands and millions of cells using single cell transcriptomics, you might get 1,000 this way in the same amount of time. Okay, very slow, but very important. So there's very characteristics of molecular characterization, of course, you might be interested in, understanding transcription factors, understanding developmental trajectories, things that are conserved in evolution, et cetera, other kind of things. And typically what's done in terms of, one of the big projects that is underway right now is a kind of attempt to understand how these cell types are kind of organized, right? And so if you have all these methods, you have the electrophysiology, you have the morphology, you have the transcriptomics, each are important and play some role in the cell's identity for sure. But the idea is that it's helpful to ground it in one technique because you can't do them all simultaneously, as I mentioned. And if you had to do it, it appears that sort of the transcriptome may be the best way to do it. And the reasons are as follows. The morphology is too variable. It's not strongly predictive of types, especially as you move away from basic types. The physiology is a little problematic because it depends on a stimulus, right? The cells only fire in certain patterns, but subject to different stimulus, you have to deal with that kind of complexity. And but the transcriptome has, in some sense, a very reproducible kind of signature, subject to all the problems of noise and laboratory experiment, of which there are many. But you can derive a kind of a base classification from that. And so most of the major projects that are underway attempt to, at least to first order, use the transcriptomics as a classification. And so this is the ones that we'll be primarily interested in. So there's this big organizations that have been going on now with the US Brain Initiative for over 10 years now. It was called the Brain Initiative Cell Atlas Network, and now it's changed its name to Brain Initiative Cell, it was called Cell Census Network. Now it's called Cell Atlas Network. The reason only is just because we've moved to include the non-human primates and humans into the picture. And so this has been the work of many laboratories across the country as well as internationally, and using all these different techniques that I mentioned to kind of survey the cell types of the brain. There's just kind of showing a kind of a map of like different labs that were involved, what techniques they use, and that's not so important to us here because we want to get to some real math, right? So we will. But anyway, this led to kind of, this led to different things. It led to a strong kind of characterization of the motor cortex, in 3 species, in mouse, marmoset, and human. It's now, we're about ready to release a very deep dive in all basal ganglia, and it's on its way to kind of, profiling kind of different things. So these are, you derive these kind of, in essence, these consensus taxonomies here, and they're often represented sort of hierarchically, as I've shown here with these kind of dendrogram thing. That's, as scientists, you have to, and modelers, scientists are modelers a lot of times, and you have to simplify. And the models, as you know, according to famous edicts, are not, they're usually wrong, right? Box's famous kind of thing, but all models are wrong, some are useful. And the same thing obviously applies here, is that, you organize cell types in this kind of hierarchical way, but in the cortex, it appears that there is a kind of hierarchical organization, but in other parts of the brain, some of the older parts, the hypothalamus, the other thing. There are many different nuclei with many functions. It's a little bit less clear if the organization is hierarchical or some more, complex graph of interactions. These are just some examples of the things. This was a study here done entirely all with the patch-seq technique, in which that Andreas Tolias and colleagues kind of, basically found all these shapes, aligned them with things, and they present them in these UMaps. And many people, I'm sure here, have certainly heard of UMaps and these techniques, and they will play a certain role in what we're about to talk about later, because we're going to find another way to look at things that may be useful. And then people derive these wiring diagrams, right? This was done by Hongwei Dong from UCLA, and this was using a kind of anterograde or retrograde tracing. You inject essentially viruses into the neuron or into the neighborhood of the neuron, and the neuron takes it up and propagates it through its kind of structure, and it, in some sense, lights it up and shows you what's connected to what. It's a very interesting technique. And then there's this, again, I mentioned the spatial transcriptomic technique. This is very important in terms of understanding the classification, because here now, when we do like single cell RNA-seq or profiling, what we're doing is we're taking single cells, we're extracting their RNA, and we're putting them through high throughput sequencing machines. And these sequence machines are able to kind of process millions of cells in the order of weeks to months kind of thing. But you lose the spatial context because you've ground everything up and you've disassociated them and sorted them. And so these techniques of these spatial transcriptomics, these in situ techniques, allow you to see exactly what cells express which genes. The difference, though, is you can't get all genes this way. You have to pick your genes. You've got to pick a panel. Whereas in the RNA-seq, it's more unbiased. You just sequence and see what comes up, right? But this is very important in the classification, because if it helps to know, these are layers of the cortex here, of the mouse shown here. Layers. All mammals have 6 layers in their cortex, more or less, and it's very helpful to know where the cell lives to do that. Okay, and there's been some nice mathematical techniques for kind of connecting these things in the absence. So we can use the patch-seq as a ground truth, and that provides us with a kind of training set to sort of associate transcriptomics to physiology to morphology. But then what we would like to do is take data sets where we didn't have that and somehow extend that. And so here's a method here which was we developed in our group a few years ago, about using kind of these kind of coupled autoencoders, where you have 2 autoencoders one trying to kind of encode the transcriptomic structure, the other trying to incorporate sort of electrophysiology, and there's an adversarial kind of contest with them. They're trying to agree on what kind of what is what, and you train this in the classical way, and you have this kind of function which is guiding how strongly they should remain coupled, and you get a really improved. You can actually extend this to bring in more data into the context, so it's quite nice that way. Other tools such as sort of variational methods and probabilistic methods through kind of things, something called ScanVI. Maybe you've heard of it if you've used these kind of tools, but sort of Bayesian kind of techniques, and it's a generalization to multimodal things called multi-VI. Come out of near Joseph's labs, and but anyway, we've used this to build an online resource where people can take their RNA-seq data, which just basically now comes as sort of sets of sequences that they've discovered, and you can map them to what cell types that we have, and it will give you back what the cell types are, okay? And so we use these kind of techniques to actually align these taxonomies, much like, so we've learned a lot about what genes are by aligning genomes of different species, okay? By aligning mouse to human, we see what are the active regions, what are the regions in a human which appear to be more strongly evolving, so-called accelerated regions, right? And you can generalize that to map cell types to cell types. If you derive these different taxonomies using these kind of techniques, you can map them together by essentially, you find markers of which kind of genes are driving the classification, and then you sort of try to align these across species, and you build these kind of consensus taxonomies. So one of the things that people do in this is, of course, is that now they're working, in the case of transcriptomics, you're working in this really, really high dimensional space, right? When we cluster these, we don't sort of usually take all genes, right? It appears that at least 80% of the genes of the genome are expressed one way or another in the human brain. And this is because the brain requires an immense amount of just basal functioning. The brain is essentially, imagine building, for as much as we would like to imagine there could be super intelligence, think of what it would take to implement a human brain in a relatively hostile environment, which has to survive and function. I mean, this is a remarkable thing of really quite an achievement for nature to do. And so what we tend to do is work with a kind of a subset of these genes which have strong differential patterns between cells, right? If we can find cells that such a gene expresses strongly and the other one doesn't, then we're more interested in that gene than those ones which are expressed ubiquitously. And so if you do that, you end up with, say, 2,000 to 5,000 genes as opposed to 20,000 or 25,000 genes. But still, that's a pretty big dimensional space. It's one of the, of course, as data analysts and mathematicians, that, the big kind of elephant in the room is that beyond a few dimensions, we really have a real hard time, working with data, right? Because of all these notions of what it means to sample in these spaces is dramatically kind of constrained. But what biologists tend to do is that they do this analysis, do this clustering, and then they project everything down into these like two-dimensional UMaps, right? Or TS&E plots, which are powerful visualization tools, but they highly distort the kind of geometry and structure, right? And I'm going to say more about what that means, but here's, for example, data that came from a profiling of an entire transcriptic profile with the whole mouse brain. So these were like sampled all over the mouse brain, right? Mouse brain is, right, it's got about 100 million neurons in it. And it's sampled quite, not anywhere near that level, but in the millions of cells. And then, you study the transcript, you classify, you group the cell types, and then you take the cells and you map them back into this UMap, and you kind of color code them based on what cell types you found. Well, and on the left, it's colored by they're colored by different levels of resolution, right? At the highest level, there might just be, excitatory, inhibitory, glial cells, okay? And then next level, you might, what we call classes, which would be more like you're getting down a little bit lower now into things that, certain kinds of inhibitory neurons or certain type of thing. And then you can go even further. And as you go further, you see the colors start to intermingle, get more and more complex and everything like that. And the question becomes, is that, how real is this, right? I mean, there is structure to it. It's not by any means random, right? It's clear that, some of this pink and areas here, my cursor shows up there, yeah. It's clear that pink goes with pink, but the local geometry is a lot more suspect. Okay, the local relationships are problematic. And it would be interesting to know that, can you devise methods to work on these classifications that would do a little bit better than that? This just shows another annotation of that. We don't have to dwell on this, but just some labels. And often what biologists will do, they'll look at this and they'll say, but what's even worse is that they will look at this and see, it's not so bad to say there's something sort of cyan colored here, and that's a different cell type because I see it looks like it's a little separated here. But it gets a little bit more problematic when they say, I see a gradient here. There's a gradient going from this to this. And the gradient implies that you have a pretty good understanding of the local structure, which these are really bad at, okay? So anyway, we put this data in, but it's still a way of visualizing and looking at things. So we put these data in our atlases and we connect them with the spatial stuff, and we make these different environments, and we build these kind of knowledge kind of things around it so people can explore it. So, and this is another level, this is another way of kind of like representing the same thing, where here we've kind of just we've taken kind of coordinates, the centroids, if you would, of gene expression, and like embedded them either using like a PCA, like a multi-dimensional scaling or something, and then there's a kind of confusion where there's a line here if there's crosstalk between these types, okay? We'll say a little bit more. So what I want to get into is some techniques for maybe trying to do this a little bit better. And so this is going to be kind of move a little bit to the tutorial thing. Excuse me if I end up, and I'm not trying to insult anybody's mathematical education here, so I'm just going to do this so that everybody kind of stays on the same page. But, you know, this is kind of in a field of interest called topological data analysis. And one of the pioneers in it was this algebraic topologist named Gunnar Carlson, a kind of strong mathematician from Stanford, did a lot of work in kind of homotopy theory. The notion is that can you apply methods of like topology, in particular like homology, right? Measuring essentially holes in objects or genus. And this notion of holes is very interesting in data because holes in data may tell you something about the structure of data. And so that's what kind of this is all about. And there's a couple different things that we want to kind of talk about here to get our handle on this. One of the things is a field called Morse theory. And Morse theory was kind of devised by this very, very brilliant mathematician named Marsden Morse, lived from 1892 to around 1997. He spent most of his life at the Institute for Advanced Study. And he basically had this kind of remarkable realization, which was really, it's a very strong thing, that essentially, that regardless, so this is in the continuous case now. We're not talking about, we're going to get to the discrete version of this in a little bit, but you need to understand a little bit about the continuous case. And so the notion is if you want to study the like topological properties of a manifold, continuous manifold, in fact, Morse's kind of realization is that if provided that I can get some kind of local differentiable structure, then if I around the critical points or around the places where this manifold really changes its kind of topology, I can actually kind of model this using a kind of essentially a quadratic form, right? I can think of it as like no matter how curvy something is, just picture something in 3D, a crazy shape. No matter how curvy or whatever it is, if it has sufficiently kind of smoothness properties, then around the critical points where it achieves local maxima, local minima, or saddles, that I can actually fit little quadratic forms, little paraboloids or hyperboloids around them to actually model the local thing there. And the important thing is that that's nice, you can do that. And in fact, what Morse showed is that there's something called a Morse lemma, that in fact, that it suffices to consider just these kind of quadratic forms here of a certain amount of positive squares. You can think of this as a paraboloid content and a hyperboloid content, if you would, something like that. And the balance of these is called the index, how many you have more of one than the other. And this allows you to fit these kind of shapes. And this is much higher dimensions. It can be done in any dimension now, but it's a three-dimensional representation. Essentially provided certain regulation kind of is kind of satisfied, then it will be okay. And the key thing is just that the Hessian, the second order matrix of derivatives, can't have any kind of singular points. It's got to be, there has to be a kind of non-zero Hessian at every point. And if you have this, then what this means is that all the critical points of the manifold, there's never any degeneracy where it goes flat or tangents can't be constructed. It's always well-defined kind of motion through a minimum, a maxima, or a saddle, okay? And then what's really cool is that what Morse showed is that in fact, these critical points, as defined by this, in fact, are very closely related to the Betti numbers of the manifold. Namely that the Betti numbers, if you don't know, I don't know if you have a course in algebraic topology, but the Betti numbers are basically just essentially counting different holes of different dimensions in the data. And that he's modeling these critical points, and the critical points are what determines where the topology changes. He's got an analytical way of modeling the critical points, and that these critical points are very closely related to the topology through these Betty numbers, okay? And so that's a really cool thing. And we're going to pursue this, but in a discrete way momentarily. So another aspect of topological data analysis is something called persistent homology. And so homology, again, if you had algebraic topologies, right, you're basically looking at certain cycles and you're kind of dividing, you're modding out by some certain kind of null cycles, and you basically find a way of capturing how loops can be made around a structure's topology in arbitrary dimensions, right? And so persistent homology is that basically what I'm going to do is that I'm going to have some way of discretizing all this, but in the same sense of like level sets, I want to be able to raise the level out of the noise and then see how the homology is changing, okay? And so typically when one does this, you build these discretizations of your space, which we'll talk about in a minute, and then you sort of study how these features persist as you go up. Here's an example right here. I'm just showing how things, this is a continuous case here. This is the classical example in Morse theory, in every single Morse theory paper you'll ever read. And I got some references for you at the end for people who want to follow up and learn a little more. But they will always show this picture. It's a torus standing up like this. And it's interesting to look is at what are the points of this? Well, if I start taking a level thing and I raise it from down here, at the very bottom it's just a point. And as I move the level up into here, in fact, I'm just there homeomorphic to a disk, right? If I cut off the whole top, I get a disk, I could push it down. But then, we're thinking like Morse, Marsden Morse right now. You can see what happens as I cross this critical point right here. There's a critical point right here where the torus kind of like opens up. And something different happens there. It changes its topology. Like Morse realized, the topology changes at the critical points. And there, it becomes homeomorphic to a tube or a cylinder if I chop off the top and just roll it out. And so people make these kind of diagrams. These are called persistence diagrams on the left here, where for each feature, I can show where was it born, at what point in time is it born as a function of raising the sea level, and at what function does it die. And that tells us quite a bit about the topology of the structure. So I raised it up. And actually, so the topology changes again. It will start to become a torus eventually, and then it'll become, you know, it'll end up being a torus at the end, right? So that's the concept of this. Whoops, we're going backwards instead of forward. So this is, you know, here's an example of just this persistence homology, something applied to data right here. You can imagine you take a bunch of points, isolated points, and these are kind of different, you're going to build up a sort of a geometric complex with this. Remember what a complex is, just a triangulation where you got to respect, your triangulation has got to respect things at the boundary. You don't have weird triangles that intersect. If they intersect, they got to share an edge. If a pyramid intersects another pyramid, it's got to share a face, right? That's a legitimate CW complex. And so you can start with data. And what I start to do is, this is either called a RIPS complex or sometimes a check complex, and I essentially start growing little spheres around the data points. And I keep track of that graph. And eventually what happens is the spheres, they start to intersect. And when they intersect, I draw an edge between them. And I keep track of that. And you can see, lo and behold, what happens right here is that at this stage in the mechanism here, sufficiently many spheres have intersected that a loop has grown in the data, and it's showing that there's a hole in the data. This is a very interesting thing. So that tells us something about the original kind of structure of this data set, that apparently it seemed to have been arranged in some kind of circular pattern, right? And so, and this will persist for a while. That's why it's called persistence. It'll persist as I grow these spheres until eventually they'll all collapse and it'll kind of merge into nothingness. But how long this thing persists is an estimative thing. So this Raoul Bott was a famous topologist from Harvard, and he had a student named Robert Foreman who basically took all this Morse machinery, and he was able to derive a kind of combinatorial analog of it that worked on discrete structures. And he was probably thinking about applications, but he himself didn't do too many applications of it. But essentially, it's non-trivial still. It's not like it doesn't really carry over as easily as you might think. But basically what you have to do is you have to define this kind of you have to work with a triangulation, and then you have to define a Morse function. Remember what a Morse function was. It was a function that for all its critical points, none of them were degenerate. And you have to kind of make an analog of that for the discrete case, which essentially amounts to assigning numbers to the edges and the faces and whatnot of the complex in such a way that magnitudes are kind of respected. In other words, if I'm an edge, then I shouldn't be lower than the value on a face too often, right? You can do it once, but not too often, etc. And then what this ends up doing, this is very powerful, because what it ends up doing, it allows us to define the analog of a gradient on this discrete structure, because the gradient is what we would have needed to model critical points on a manifold. If we want to do the same thing on a kind of discrete structure, I've got to have some notion of gradient, a discrete analog of it. And that's what this does. It allows me to construct a discrete gradient field, which now I can describe what are critical points in this, and I can actually build it up. And so that's what this discrete Morse theory is all about. And the critical cells will correspond to minima, the critical one cells will correspond to saddles, etc., just in the same way as in the continuous case. And there's an analog of the continuous case, which says that discrete Morse function is equivalent in a homotopy sense. You can deform it to your complex consisting only of critical cells. So I can reduce this through some operations of merging and dropping faces and whatnot to greatly simplify this, okay? And this slide here just presents, kind of some, the analogs of what Morse theory and what persistent homology are and how the two of them are useful. So a natural application of this is actually, it was done here by my colleague, who I know some people in the audience here know, Partha Mitra. This was applied to in a comparatively more pragmatic case, I wouldn't say easy, of reconstructing neuronal shape. And what you do is the analogy is that you imagine that the neuron and the intensity in the measured image is a kind of height function. So the Morse function becomes the intensity in the image. And by using this technique, I can actually, so why would you use it? Well, the problem is you use it is when you do light microscopy in particular, you try to image something and you get, because of the nature of the light and the imaging and the sectioning, you get breaks that sometimes with the eye will look at and you say, oh, it's obvious the neuron goes here. But when you apply classical image processing algorithms, and classical image processing algorithms used what they were called morphological operations, interestingly enough. And they meant things like, the classical morphological operations of image processing were, well, take the intersection with this, complement this, level set this, etc. These all things. And they have proven to be, there was thought before, years ago, that such a calculus of these operations would have enabled us to solve very complex image processing problems. Today, we realize that image processing problems are much better solved by deep learning approaches and these kind of things. And these morphological operations have become somewhat, I would say, disfavored in comparison. But what we've done here is he's combined the kind of a UNet kind of and various kind of machine learning methods by a seed generated through this kind of a discrete Morse theory, and greatly improves kind of the technique of segmentation technique. And basically, you want to think of it as that, again, the analogy here, because we'll want to use this analogy as we move into the transcriptomic realm, is that this is truly a 3D structure here. The height is literally the intensity of the neuron measured in a 2D slice, right? And so you can reconstruct it in that way. Some results can be done in 3D using the kind of various other methods that involve clearing the brain and stuff like that. And actually, Edvard Moser and May-Britt Moser, the famous Nobelists who studied grid cells and place cells, they have actually used these techniques to model data and show that in the hippocampus that kind of the response patterns to, and certain things in the grid cells were actually kind of formed a kind of toroidal kind of manifold, and they reconstructed that by moving their data using the this Morse kind of topological data analysis into that. So what we've done is that we've tried to adapt these methods to work in much higher dimensions. And again, we're going to move up into hundreds of dimensions, and there will be inaccuracies, right? But the hope is to try to provide more, at least some more local accuracy than you get by massive dimension collapse ab initio and doing that. And we'd like to do better even than, say, doing PCA down to 100 dimensions and then doing the results. So we built this out. This was done by a woman who's a great developer. She's really the master of topological computational algorithms from UCSD. This was a student of hers, Lucas McGee, that we worked together. And he kind of basically, they devised these kind of discrete Morse algorithms. And I can't go into the details, but essentially through some kind of complicated graph theoretic options, found ways to simplify the techniques and build up these data structures in ways that could be processed more efficiently. It's still not terribly efficient, okay? We're still talking about the whole running time is worse than quadratic still, okay? But it's manageable for some kinds of problems that I want to tell you about. And the overall algorithm structure, without going into the details of what was done to improve it, are just, you essentially, you have to build up this filtration. And a filtration, if you ever had like measure theory or you studied probability or stochastic processes, you remember that a filtration is just essentially, it's a way of organizing data in such a way that I keep on either adding structure or taking away structure in a hierarchical way. I improve the resolution, right? Think of it as ways of kind of, you learn more. The classical way it's used in stochastic processes is as you learn more, you change the level of knowledge in the filtration. And for our triangulation, it's how densely things are kind of triangulated. And then we build up these discrete Morse things on this thing, right? And then as the Morse theory tells us, you can find, even though this dimensional space is very high, to understand the critical points and where they change, you only have to look at these one-dimensional manifolds in this high-dimensional space. So just like in 3D, where I can walk, if I really want to know where the problems are when I go hiking, I just have to follow the gradient up and down ridges in between peaks and valleys and saddle points. You can do the same thing in these high dimensions by extracting this one-dimensional manifold from this Morse structure, okay? And so these are the kind of things, so using this, we want to look at cell types from this Morse perspective. And so this is kind of a critical, we've arrived at a point of kind of a critical juncture here. And so we want to think of cell types, at least transcriptomic cell types, all right? So I'm only talking about the transcriptomic scaffold here. I'm not talking about putting on the physiology and the morphology yet, but just this transcriptomic thing, although you could mill a space out of all of them. And so the premise is that if two cell, anyone who works in the classification of cell types uses this premise, that if two cells express the same genes in the same way, then they probably should be considered the same type, right? And so essentially what we're going to do here is we want a measure that says how closely we should associate cells, and then we're going to triangulate our space and build up this complex based on these relationships of proximity, and then we're going to use the Morse theory on that, and we're going to use the persistent homology on that, okay? So the idea is that we'll have a notion of gradient on this, and we'll be able to change the resolution to see what features matter. In other words, what a type will become stronger if I can raise the persistent level and it still remains, okay? And so from a Morse perspective, two cell types are distinct if there are relative maxima in this space, and if they can be separated by some meaningful drop, okay? And it turns out that we have found that the best measurement for similarity is basically a Jacquard index between two cells. So two cells, what they do is that they look at all their neighbors in the sense, and you can use kind of correlation of the expression vectors. You can use information, whatever you want. But the key is that you don't use those raw measurements, but you smooth those raw measurements by only essentially saying, I'm like you if we have the same friends, okay? And that's your notion of proximity. And that essentially builds communities nicely by that kind of metric. And so that's our notion of similarity. And then we build up the gradient structure on that, and we build up this thing. So just, I know this is kind of a mouthful, but if you think about this just keep this 3D analogy in mind, this is what's going on using all this machinery in a high-dimensional space. So that's that. That's the algorithm. You start by building this kind of complex. You build up the whole thing. We don't have to go. I think I've intuitively given the idea now. But let's apply it now and see what happens. So, one of the things we did, we studied this kind of this data set here, which was, this was a few years ago. It was an entire kind of transcriptomic profiling of all of the cortex and the hippocampus in the mouse. It's 1.3 million cells. They were assigned to different levels of resolution, class, subclass, supertype, and cell type. It came from 18 different regions, and so we used the top 2,000 to 4,000 genes, essentially depending on what resolution we wanted, to sort of build up this kind of complex for. And so the first thing we did is we went and studied and say, if we built this discrete structure up, did it respect local structure better than the UMAP, right? And to do we did this, and we studied this, and it does. And we found, like others have found, that the global structure is well preserved in the UMAP, but locally things are just messy, a mess, and it doesn't describe well. And so there's lots of validation here that went into kind of showing that the structures that you build up are valid, that they in fact span the space of interest, and that they're robust to certain parameters you have to choose because when you build this up, you got to choose how many neighbors you're going to count to call something similar, and so it should work over a range of those, whatnot, and all that kind of thing. And so there's various validation steps that one does. But even here's a good example right here. These were two types here. These were identified cell types in the data that they found. These are transcriptomic identified sets. And what we did is we build up the thing, and then we follow our one-dimensional path in this manifold the Morse structure that we build up. And here I'm just plotting their kind of Jacquard index, or how similar one is to the next as I moved on this path. So this is kind of a critical path. It's a path of greatest change in this transcriptomic space. And it goes down, and it achieves, this is a saddle kind of right here, approximately at the minimum it would seem. And it goes from peaks down to saddles, back up to peaks. And so here's an example here. This was a UMAP here showing the colored orange and colored brown of the two different cell types labeled somatostatin 88 and 91, and I've put the Morse path over it. And you can see the Morse path does eventually jump from one to the other, but it does all this noisy moving around back and forth in each side before it jumps, And so that's the problem. And anyway, if you study this, you can see that in fact, that if I look at the genes that they used to drive this change, and I plot them, that in fact, I see that they're expressing high in one cell type, and that they cross pretty much exactly at that saddle point, that minimum point. And this suggests that it's kind of a sanity check that in fact these are kind of working pretty well. So we're running out of time a little bit, and there were other things we studied. One of the methods that's really powerful is studying, I mentioned, these gradients. And we can study, exactly how smoothly transcriptomic changes change as you move across a data set, much better than these low-dimensional methods. But I don't want to say, for example, that this is going to replace clustering of large-scale data, of transcriptomic data. but it provides a way of understanding better the local relationships and the gradient relationships in these data sets. So I know I hate it when people skip through slides that they didn't prepare for, but I spent more slide time in the beginning and in the end, so you'll have to excuse me. But in any way, what we did here is that, so this is an example here. This is showing essentially, these are the GABAergic kind of cell types in the cortex and the hippocampus of the type, the somatostatin type. Somatostatin is a kind of neuropeptide expressing gene, and we looked at all the cells that were expressing that. And essentially, using this Morse structure, you build up something we can call a Morse persistence taxonomy. And that just says this, is that it says, I find the local peaks and whatnot, I call them, and then I keep raising this threshold, and I do the same thing. And the color gets hotter if it survives a higher and higher threshold. So therefore, hotter should be interpreted as cell types more similar to each other. Blank should be unrelated. And it very much, and then we redrew these kind of constellation maps from the original paper, and you can see that in fact it was quite similar to what they did, but we have some discrepancies that we found. That there are some things that they claimed were there that we found were a little bit kind of off, and so we reclassified. And then here we were able to do the thing for brain-wide, for all inhibitory neurons brain-wide, 177,000 cells. And this, it actually, at a level just above the lowest level, it really kind of validates these as very, very kind of robust types that actually survive very well. So I think that the main value here is, again, not so much in de novo clustering, but in really kind of validating kind of clustering structure and more accurately describing its local properties. So I'm gonna stop here because maybe we want some time for questions. I think we're at time. But I made there's some topics for further study. These slides actually we're going to make available for people, I guess, yeah. But also I made a reading list here if anyone would like to, if you'd like to learn more about this. These are some key kind of papers, more or less from a tutorial standpoint, that will introduce you to Morse theory, topological data analysis, and discrete Morse theory and these things. Check out, if you read these papers, you'll know more than I do. So check them out. Thank you.
Okay, questions?
Hi, thank you for this awesome talk, Michael. And I have a question on slide 18, when you were using the classification of the brain region of mice. So is there any predetermined subclass number or class number that we use where we'll cut down the classification, or so this is my first question.
Sure, could you repeat that question?
Yeah, sure. When we are doing the classification, when do we think, like, how many separate classification we want from any classifier? Like, for example,
how many clusters?
Yeah, how many clusters?
How do you know when to stop?
Yeah.
Yeah. So, like in all data analysis problems, that's an issue, right? And you have to build some kind of statistical criteria for what's essentially robust enough. And there's various ways you can do that. One is you can compare it to some other truth. The second way is you could as you raise your threshold, you can certainly order the features from the most robust to the less robust. So for us in this framework, say for cell types, something is going to survive very high for one of two reasons. Either there's a lot of cells of that type, or that type is very isolated from other types. And so, in other words, it could be a type with not so many cells, but it's think of it as like a tall, that would be like a tall, thin peak, essentially, which drops off like, some kind of like a spire or something. Or it could be a broad mountain thing that has a sufficient dip, but is expressing many cells, and there are many cells that do that. And so you just have to use statistical measures to understand where to stop. Yeah.
And also in the Vietri-Rees complex, like when we had the persistent homology and the Betti numbers, so usually in algebraic topology, one of the key features that we use is to compare between two different spaces, where like, so for example, two different spaces, if they are homeomorphic or not. So here I understood like we're using the persistent homology, the homology group or the Betti numbers to think whether two regions, how they behave in terms of like how much persistence they are if we increase the level of persistence. But here, one of the key things that you are using in the Morse theory is the underlying topology, is the differential topology, differential manifolds. What will happen is if it is not locally differentiable, are we using some smoothing techniques there?
Yeah, that's a good question. So that's a good point. The point he's making is that there's really no guarantees that the Morse actual in the requirement for the Morse case, right? That that this Hessian is non-vanishing in each point. That this probably doesn't happen in real data. There's going to be degeneracies, and how do you account for that? And I don't think we have a good way to do that. We don't have a good way to do that just yet. We can do it. The way that we're looking at it is essentially that the results, seem to be holding up under essentially, subsampling and redoing it, then it's apparently robust enough. But that's a problem with the whole paradigm a little bit, that you don't have a firm guarantee that this kind of continuity conditions are really available in raw data. But, and you could smooth, but the smoothing is, you got to be careful with that, because then you end up smoothing away too much of the data. So another way to look at it, of course, is somewhat like stochastic gradient descent, that it's unlikely that if I do this, kind of randomly, I'm going to hit things that are perfectly vanishing. But it's a good point you raise, yeah.
Thank you for a fantastic talk. My question is one of scaling. So different genes like to express at wildly different magnitudes, and it is hotly debated in some communities whether to regularize and normalize them at all. My understanding of persistent homology with the epsilon balls is that distances in this high-dimensional space matters. Is the Jacquard index that you talked about a way to kind of get around that? What are some ways that you can envision that either due to our incorrect perception of the right distance to use, the right normalization to use, or the right way to pick neighbors might give rise to false topologies that don't really exist from the viewpoint of the cell?
Also good points. And from there, we try to pick, that we work with the most robust genes we can find in order to get, kind of differential patternings which are going to point out features as good as possible. But some of these other points you're mentioning are technicalities that you have to worry about, and some of them are still a work in progress, I would say. But
hi, I have one question about the use of the DM graph reconstruction algorithm for tracing neurons. What will happen if you have an intersection or a crossing point? Because I can imagine if you have a single neuron.
Intersections. Yeah. Well, it's not going to resolve that. And that's you're not going to, if the intersection is at the same intensity, which they may not, often it's not, right? But if the intersection is at the same intensity, you're going to have a problem potentially by any technique. This technique is not, it's not, I don't know. If you were following the gradient, you would probably keep going straight at the intersection, but I'm not quite sure there. I think that, for example, in like now, even we use, a lot of deep learning techniques to segment electron microscopy data, right? And so this is proven to be, electron microscopy data is particularly problematic because in a given section, to the eye, except for the highly trained eye, it's almost meaningless. You can't really see much structure there unless you're an EM kind of, person. And the problem there is also if you miss a section, one or two sections, you're dead, because it's how something moves between there. And so these deep learning methods have been very, Sebastian Sung's group and others have at Princeton have actually found good ways to do this, but it all still requires substantial proofreading afterwards to actually validate it. And so the proofreading has not been gotten around. I think probably the same thing would be there.